Hello and welcome everyone to this edition of the ACTnet Asia Pacific Regional TB Journal Club. Today's 10th of September 2025. My name's Harry Walker. I'm an ID physician or infectious diseases physician and microbiologist. I'm currently on Ngunnawal country in Canberra. I'm on my way up to central Queensland where I'll be working as an ID physician and microbiologist there. I'd like to acknowledge the traditional owners of here in Ngunnawal country in Canberra, but of course of all First Nations people in Australia and indeed throughout the Asia Pacific. We're in for a real treat today. I'll just hand over to my co-host Gail Cross to introduce our presenter.
Hi, everyone. Thanks for coming today. I can't quite see how many people we've got, but like Harry, I'm very excited about this presentation on bioaerosols and TB. My name is Gail. I work at Monash. I'm an ID physician and a clinician researcher based here in Melbourne. And it is with great pleasure that we have Professor Helen Cox, who is my colleague now at Burnet, but Helen is a public health practitioner and an epidemiologist who has really dedicated her life to TB. So Helen's from Australia, but she went to South Africa, where she worked at the University of Cape Town for many years and has led a lot of studies in TB that have been really changed the way that we think about the epidemiology of TB and the treatment of TB as well. She's won grants from the Wellcome Trust, from the Gates Foundation, from the EDCTP, and is a real TB expert. So we're very lucky to have her at Burnet and also lucky to have her presenting on the topic of bioaerosols and TB. So I'll hand over to her.
So I would just like to add my acknowledgement of country as well. I'm on the lands of the Wadawurrung people. I'm in Geelong in Victoria. And I pay my respects to elders past, present, and emerging. So these are the two papers that I wanted to present. There are two because they're very closely related. They were published last year in February and September. And they're from a group of researchers at the University of Cape Town in South Africa, where I was for a long time. I wasn't involved in these studies. I just know the people reasonably well. But I think they're really quite fascinating and potentially change the way we think about TB transmission. So I'll start with a bit of background. And there's not a lot of background because we're all working in TB, so we know about TB. But essentially, this is what we think we know about TB transmission. So we've always understood that people with active symptomatic TB are the primary drivers of TB transmission. And these are the people who are infectious and who we need to identify and treat to stop transmission. We also have this notion that TB transmission is primarily driven by coughing. So expulsion of TB bacilli through coughing. And it's often coughing patients who are thought of as being infectious. And this is the basis behind cough triage and all sorts of various interventions in the past, which have not been incredibly effective. We also think that the greater the bacterial burden in sputum, the greater the transmission risk. And, bacterial burden in sputum measured by smear microscopy or culture or expert, this is how we gauge transmission risk. And then we also think that 2 weeks of standard chemotherapy is sufficient to dramatically reduce the risk of TB transmission. And there is some evidence around some of these notions, but actually when you start to explore them, some of them are not very well evidence-based. So aerosols come from peripheral lung fluid, aerosols that we breathe out, which are in the terminal bronchioles for the respiratory physicians, of which I'm not one. But sputum is actually produced in the sort of upper conducting airway. So it's a different location. And of course, it's a different medium for holding TB bacilli. But as bioaerosols are actually what we breathe out, they may be a better sample to assess TB transmission risk. But of course, the problem has been that we haven't been able to measure TB in air very well at all in the past, if at all. And then the third point that is really important is that we now know that around half of all the active TB from TB prevalence surveys is actually asymptomatic. And in most high TB burden settings, this half of the TB burden is predominantly not diagnosed or treated unless you have active case finding that is independent of symptoms. And of course, this, relates to our better understanding of this spectrum of TB infection and disease through what we used to call latent TB infection through to active TB, and how this is now a progression and a spectrum rather than a binary dichotomy. So the aims of these two studies were to investigate viable MTB release at baseline 2 weeks, 2 months, and 6 months in various participant groups by measuring MTB in breathed out aerosols. And the three groups were lab confirmed, so expert positive, TB positive individuals, expert negative individuals, but those who are clinically diagnosed with TB, and a third group where there was no TB diagnosed, so no TB treatment after screening for TB. So that was the first study. And then the second study was to also investigate bioaerosol release, but looking at a sample of community participants, so not people from being investigated for TB in clinics. And in the second study, there was repeat sampling over 2 months. So these studies were conducted in Cape Town, which has an incredibly high TB prevalence. And in particular, these studies were conducted in a relatively small township called Masapumalele, which has a population of around 25,000. It is very densely populated. You can see here, most people live in shacks. They're very close together. They often flood. There are often fires, which are with tragic consequences. And there is a very high TB prevalence, a very high HIV prevalence as well in this setting. So TB case notification is probably more than 1,000 per 100,000 per year. And that's just case notification. Prevalence or incidence is probably an order of magnitude, double that at least. So in this township, there is a dedicated facility called the Aerobiology Research Unit that's part of the University of Cape Town, and that's where these experiments were conducted. So aerosol sampling is quite complex for any pathogens, but particularly so for TB. So in these studies, there is a sort of a chamber that a participant will sit in a Tyvek suit to try and reduce the risk of contamination. And they'll breathe into this cone, which will collect the bioaerosols. And part of the system includes measuring temperature and humidity, CO2, and counting particles. So it's a fairly sophisticated setup that has been established over a number of years with a lot of trial and error to get this to work. So within this chamber, the participant can do all sorts of respiratory maneuvers, and in particular for these studies, the ones that are relevant are sort of forced vital capacity, so forced breathing out as much as possible, normal tidal breathing, or voluntary cough. how to identify the TB bacteria? Well, this is the complicated part of it. So the captured aerosols are stained with a particular stain that has to be taken up into the cell, and then you can concentrate the samples and then view the sample under a fluorescent microscope to look at the fluorescence from the stain. Now, it's important that the stain is only actually taken up after incubation into viable cells. So this is a way of actually measuring viability as well as the presence of bacteria, which is important. So they also attempt to identify MTB through a process called droplet digital PCR-based molecular assays, which the microbiologists probably know much more than I do. It's quite complex, but this is a PCR-based assay looking at target DNA. And then also various other staining techniques of auramine and Z-Nielsen staining. So this whole procedure is explained in an earlier paper. So the first study onto the results aimed to look at these three groups of participants: the lab-confirmed TB, the clinical TB, and no TB, not treated. So firstly, they pretty much all had symptoms. A few in the clinical TB and the not TB group didn't have symptoms. These are probably contacts that were screened as part of the contact investigation process. But mostly this was a symptomatic cohort. And then if you look at the sort of bioaerosol TB positive, this was the astounding sort of finding that jumps out of you straight away, is that all three groups had very high levels of MTB detected in bioaerosols. Indeed, across all three groups, more than 90% had bioaerosols detected, and there was no difference across the three groups. So those diagnosed with TB and those not supposedly diagnosed with TB. Importantly, there wasn't really any correlation with any of the clinical or demographic factors, including some of the pretty obvious ones that you would think of, like previous TB, HIV status in this setting. So then they looked at the sort of follow-up tests over time. So at the 2-week mark, 2 months and 6 months. And you can see that when you looked at the sort of the collective percentage that was still positive, this did go down in all three groups, but similarly, it went down in all three groups. And just to put it as an aside, all of the TB symptoms, these were primarily symptomatic people, also declined over the same time course. And I've put a note at the bottom here that unfortunately they were not really able to culture very many of these aerosol samples at all. A very few were. And mostly this is probably down to the fact that there is just not enough bacteria there to be able to culture effectively. And so they also, this is just showing what we saw before. There was no impact of TB treatment, HIV, or previous TB treatment on this sort of decline in the proportion that were bioaerosol positive. So this panel A is all of the participants, and you can see that it does decline pretty much all the way through, but there was no difference by previous TB treatment status, whether they were treated or not, or by HIV status. So the key findings of this one. So MTB was detected in a very large majority of all the three participant groups, including those not diagnosed with TB and not treated. And I guess, we can perhaps postulate this is transient, spontaneously resolving MTB, infection. And I guess this is why I mentioned at the start that this is a very high TB prevalence setting. So the force of transmission is incredibly high here. There is TB everywhere. And then both the proportion of participants with MTB positive aerosols and the number of MTB bacilli identified declined over 6 months at similar rates, irrespective of TB treatment. So there is a decline over time, but a proportion of all participants in the groups remained or became MTB positive at 6 months. So there was a group who weren't at the start, who then actually became later on. So, you know, is this reflecting a sort of non-sterilizing cure? So we may have cured some people, but we are still left with residual bacteria there. MTB detection was confirmed by the ddPCR technique, and the majority of the aerosols were also positive on auramine staining, but not on the Z-Nielsen staining. And this in itself suggests that the mycobacteria are in a different physiological state in the aerosolized, in aerosols, compared to what they are in sputum, where they're obviously very easy to stain on with Z-Nielsen. I've said insufficient DNA here, so it's not really possible to do a lot of DNA techniques on the aerosol on the aerosol samples, except for the ddPCR. And whether this is poorly culturable bacilli in terms of physiology, or if there's just not enough bacteria there, is an open question, really. So, study 2. So these are community participants. So there were 2 cohorts. The first was a cross-sectional survey with a random selection of sort of blocks of land, so shacks in Massapumaleli, where they just did a screening visit, and they looked at these 3 manoeuvres. So the first, forced vital capacity, tidal breathing, and cough. And they did a follow-up visit for quantiferon gold and gene expert. And the second cohort was more longitudinal, so they wanted to do serial sampling over 2 months of a cohort. So the effect of the 3 different respiratory manoeuvres, I think, is quite interesting. So this is the percent that show bioaerosol positive. So the green is the bioaerosol positive for TB, and it's very similar for all 3 manoeuvres. And this is showing the DMN, the count of particles based on staining when you see that, and also very similar across all 3 manoeuvres. So there was no difference. Overall, 80% of participants produced at least 1 MTB positive sample. So these are community participants, not in a primary care clinic and not being investigated for TB. So they actually ended up dropping cough from the cohort 2 findings. And actually, in a separate study that used some of this data, they estimate that only between 3 and 7% of the total bacilli, MTB bacilli bacteria released per day is due to coughing. And this is also based on measuring cough frequency using measuring devices. So it's not induced coughing, it's just spontaneous coughing. Whereas most is actually due to normal tidal breathing, which is not surprising when you consider the amount of time that you would be coughing and normal breathing. So this was also important. So this graph shows the sort of within individual variation and between individual variation in bioaerosol MTB production. So along this graph, these are each of the participants. And of course, there are some who are not bioaerosol positive, some that are very low in terms of counts that you can measure, and some which have much higher counts. But when you do two measurements from the same participant, there's actually a very high correlation. So these error bars reflect the two measurements for each participant, and they correlate very well. So there's quite a good consistency between these two samples. And indeed, most of them differed by less than 5 bacteria. So I put this in factors associated with TB positive bioaerosol, and essentially none of them are significant. I mean, these are all important factors that you think might be important in terms of age, biological sex, previous TB, of course, because we still think that there are dead bacilli that might be expelled. Quantiferon gold status as some measure of TB infection status, and of course HIV. But none of them are significant, but this really is a relatively small sample size, so they may well become significant if they were to measure more people, which is immensely time-consuming and expensive to do, but I think they are in the process. So what happens over time? So these are the 3 visits, so at 2 weeks, and so baseline 2 weeks and 2 months, and there's no significant difference between the percent that are bioaerosol positive over these 3 visits. But actually, when you look at individuals, they vary quite a lot. So some stay kind of about the same, either with no bioaerosol or low level. Some sort of start low and go high, and some are all different combinations of the above. Some are, basically there's a wide range of changes within individuals. So, questions and implications. I think the first question that people really ask is this real? Does this really reflect what is happening? Are these bioaerosol measurements accurate? Are we really seeing viable MTB? And there's been a number of studies now published from this group which have described quite detailed validation of the methods that they've used. So there is strong support for that. The viability of the bacteria has been confirmed by different methods. So that's important. And I think what is also important is the consistent results between the same individuals with repeat measurements on different days. And a key question is, of course, where do the bacilli come from? You know, is this some form of immune escape? And Gail and the other immunologists, Manu, could probably postulate on this better than I can. I think another key question is really, are these bacteria in bioaerosols able to cause infection? They're clearly most likely in a different metabolic state to that in sputum, but they are the MTB that is being breathed out, and we know that transmission is caused by rebreathing air from people who have TB. And certainly the older guinea pig models where you get guinea pig infection from people who have TB, we know that breathing out air from if you have TB is liable to cause transmission. And then we have a cough, particularly not a major driver of TB transmission. And I think we're beginning to realize this now. Cough is probably an indicator of disease severity and pathology, but it may not be the main driver of TB transmission. And then can we identify people with high bioaerosol release? And what does this mean in terms of TB transmission? And I think this last point really is TB disease required for bioaerosol generation and therefore transmission? So based on this last point, this is, I'm just checking the time. It's possible that people are actually moving sort of constantly back and forth across this TB spectrum that I showed at the start of this presentation. So, this is a paper that was published in 2018, but I think it's got a quite nice diagram here where a person may get infected. We know there are some people who rapidly develop active symptomatic TB. We know that there are some people who just who don't and stay as what we used to call latent TB for long periods of time. Some people probably clear their infection, some rapidly reactivate, but some also may do this sort of cyclical TB disease where they have symptoms for a little while and then their immune system may control that for a little while and they stay in that sort of state. So potentially that is what we're seeing. So, people moving back and forth across this spectrum of TB. And indeed, there was a modelling study done recently. If you took a sort of a clinical cohort of people who we know have TB and you looked at their outcomes over 5 years, we know that without any intervention, we know that some will self-recover, but nearly a half will probably die of their TB. Some will stay maybe in this transitional state and some as sort of this subclinical TB and a reasonably large group with what we might call minimal TB disease. So this is the clinical cohort that we have kind of always thought of in terms of TB, but then there's probably also a subclinical cohort who also have a risk of rapid progression and death, but a much larger proportion will stay with minimal TB disease and maybe a larger proportion who will recover or stay in this transitional state. So potentially that might be what's happening. Overall, I think this is an area where we desperately need more research to really understand TB transmission. And the key reason is around, well, what do we need to do for TB elimination? Because that's really what we need to do moving forward. We can't just keep doing, let's just diagnose who's got active TB on what we used to think and then treat them, because we're never going to reduce the burden in most of the high TB burden settings by doing that. So this group have now developed a portable RASC, respiratory air sampling chamber, which I was trying to get a photo of and I haven't managed to get one yet. But it would be really good to start using this in different settings to try and see whether the phenomenon that is seen in Cape Town is seen elsewhere, and if we can really try and define, TB transmission risk a little bit more. So I think that's all I have. And I know that was quite quick, but I would be, I think it prompts a lot of discussion. So it would be really good to
it certainly does.
People have to say.
Thanks a lot for that. I'm sure I'm not the only one who's feeling like this has turned upside down a lot of our understanding of TB transmission. And yeah, I might start with some of the discussion that just after you described study 1, there were some questions in the chat really kind of probed about the specificity of the tests that were used. So Kira Chua asked about how specific the Triolose test is for Mycobacterium tuberculosis. And what was the rationale from the authors or otherwise about the drop in the level of detected TB in the patients that didn't have a diagnosis of TB through other methods?
Yeah, so it is quite specific. It is specific for MTB, the DMN. I've forgotten what the DMN stands for now. Trehalose fluorescent probe. It's a probe that is specific for MTB, so it's only taken up into MTB bacteria. So it is specific. So what's the biological rationale? I think that's one of the open questions, really, because it makes more sense in the second study that you would get people who have bioaerosol positive detected and then don't over time and then go back and forth. But when in the first study, when you see that drop across 6 months, and it was so high at the start, it's possibly related to the fact that the reason why they were screened for TB in the first place. So,
right.
I mean, they all had symptoms at that point. And of course, the community specimen sample didn't have symptoms. So that would explain why they had such high positivity at the start, and they all declined because all of their symptoms declined, even the group that were not treated for TB.
So group C in study 1 was people who had symptoms compatible, but presumably didn't have a typical X-ray, didn't have enough features that a clinician would say, look, this is clearly TB, even if the tests are negative. It was...
and to note in this setting, of course, there is a lot of clinical TB diagnosis because it's such a high HIV prevalence setting. So clinicians are actually very good at diagnosing TB clinically.
Wow.
Yeah.
It's so interesting, to me at least, that the level of how much TB you find in the aerosols drops similar whether you treat them or not. Because presumably Group C, or did Group C receive any TB treatment on the basis?
No, none at all.
No, they didn't. No. Wow. Yeah.
Yeah. Well, I mean, it's possible that had another sort of respiratory infection, which then, prompts coughing and, changes, lung pathology changes, which, if there is TB bacilli present, would increase bioaerosol positivity. But I am completely speculating because I just don't think we know.
One of the things that came to mind when I saw this, when you, as you presented it, Helen, was just whether we are sampling a space that we've never sampled before, right? We all the studies that we've done, very few of which have been bioaerosol, are really sampling sputa. So sputa against trachea, this is really sampling the air that's present within the airways. And, the thing is, maybe we have a very poor understanding of the fact that in a high endemic setting, people are breathing in and out TB all the time. It doesn't necessarily mean that they are infected with TB, but it is certainly in their airways. And perhaps their immune function in their mucosal systems actually prevents them from becoming infected. So I think Carrie's question about whether they mount an immune response, what their quantiferon is like, clearly there's a population of them that do have TB, clinical TB, as we call it now, meaning they're infected with pathology in their lung and, sputa that is microbiologically positive. But really, is this, I think your question at the end was, is this viable bacteria? Is this bacilli that are transmissible? Those are questions that are front and center of my mind. And I think that the hint is in the fact that there is something different about these bacilli compared to the others. The fact that they, don't stain normally, they only take up the auramine stain. My understanding, Kyra, of trehalose dimonosaccharide is that the reason that they use it, at least in cell-based studies that I'm more familiar with, is that it's actively uptaken into the cell. So that suggests that there's some metabolic activity of these bacilli. So I don't think this is like, dead bacilli, but there certainly are bacilli that have got a dormancy phenotype. So they're dormant, but they're somewhat metabolically active as well, but certainly not the same as active replicating bacilli that cause disease. I think that's kind of like a very poor scientific summary of those changes. Anyway, I better not carry on for too long because there's some really good questions here.
There's a question on, could you recap who the 3 groups represented? So, so these are all people who were being, in the first study, they're all people who were being investigated for TB in a primary care clinic as per standard protocols. And primarily the reason is presenting with symptoms, although there are some that didn't present with symptoms and they would have been screened for other reasons, close contact of a known TB patient. And so the first group, Gene Expert is the primary means of diagnosis in South Africa. So they were Gene Expert positive, and obviously laboratory confirmed TB then, pulmonary TB. The second group were Gene Expert negative, but a clinician decided that they probably did have TB, and therefore they were placed on TB treatment, clinically diagnosed TB. And the third group were people who were investigated for TB, were Gene Expert negative, and a clinician decided that they didn't have TB, so they were not treated for TB.
Helen, was there any group crossover? So say if someone was group C and then their symptoms continued and in 3 weeks' time they had a positive sputum, they were reinvestigated, would they be removed? So would they be not seen in group C in the 6-month follow-up?
I'm just trying to remember. I think there were, when I've spoken to one of these investigators before, there were a couple of people who were in that category. Who initially were not diagnosed with TB, and then when they represented, they gave another sputum sample, which then came back as positive. I think they were removed from those groups. Yeah,
I'm just thinking similar to the comment that Kira added, that she's saying that if we accept that the bacterial load waxes and wanes over time, and especially without treatment, like group C in study 1, that it just doesn't make sense that the bacterial load in the aerosol would drop so consistently and dramatically. And I think I'd add to that, it's hard why does it drop seemingly to the same extent as treated people? Because then, it seems that the studies implying symptoms go away without treatment is the same drop in TB in the bioaerosols as treated for TB, which I suppose could be true. It's pretty extraordinary.
Obviously you can't look at a group who we know have TB and not treat them.
Of course, of course. Yeah.
But it's possible. I'm sure that group would not decline. No, I'm not sure, but that would be the hypothesis, that they would not decline over the 6 months.
Yeah. Well, so I suppose it does make a bit more sense if it's people who had symptoms and their symptoms do resolve and they don't develop clear TB disease, then I suppose it does make sense that without treatment
although the second study, the Cell paper, sort of suggests that, it's not a matter of coughing. It's not a matter of your symptom burden, right? I mean, whether if you're tidal breathing or you're speaking or you're breathing, thing is the same. So really, I think this brings up a lot of questions, more so about, do we really understand what this space is that we're investigating, and what does that mean? Katie had a question that came up in my as well. Katie Dale, she asks about study 1. Was it possible that the sampling equipment was actually contaminated, and that might be the reason we're seeing both such consistent levels of bioaerosol TB presence across the 3 groups, but also that consistent drop over time? Because you'd expect that if this was an equipment issue, you would see the same data as we did.
No, so I don't believe it is contamination of the equipment. They did a lot of blank runs and all sorts of things in between, both earlier on as part of the validation of the method, and within this study. And of course, these participants were not all followed at exactly the same time. So, some people would have been being tested for their 6 months, while some people were in their first, baseline measurement. So it wasn't all temporally tested at the same time. But I don't believe it was. It is contamination of the equipment. The reason I put the last slide about the, doing this elsewhere is I think it would be really interesting to do this. I mean, there are people with TB in Australia, as most of you know, so we could look at TB bacteria in bioaerosols from these people, but also in populations which are not exposed to so much TB around. There are obviously household contacts of those who are diagnosed in our setting here, but most of the population are not. So I think that would be really interesting to do, also in relation to IGRA positive and IGRA negative groups, etc., to really try and get a handle on some of this work.
In the discussions, Helen, from these two papers, as someone has asked, really, was there any comment made on the viability of these organisms? And rather than viability, I think maybe the transmissibility or the infectiousness of this. And then maybe after you answer that question, we could go to Rick.
Yeah. So, I think viability and transmissibility are probably different things.
Yeah.
Viability, we know that there are, very slowly replicating TB bacteria. And so it's quite hard to culture them. In fact, it's really hard to culture them, but they are viable and you can reactivate them with various laboratory techniques. So they're just sort of in a different metabolic state. In terms of transmissibility, it's incredibly hard to know how to measure that. Obviously, we were talking before we started about human challenge experiments for flu. You can't do that for TB.
Well,
yeah, it's an interesting question, isn't it?
Well, can't we?
No, we can't. And, the only model that we've had is the guinea pig model to look at infection. Excuse me. But guinea pigs are incredibly susceptible to TB. And I'm not sure that, it's not the same as a human model for TB. Yeah. So I don't know. I think that is a key question. How do we actually measure transmissibility? Yeah.
Let's go to Richard Turner, who's got his hand up.
Yeah, hi there. It's a fascinating topic. I missed the first part of this meeting, unfortunately. I think the Zoom link was incorrect, but I actually, my PhD was looking at cough and TB transmission back in London. And yeah, as everyone said, this kind of mind-blowing kind of, completely new way of looking at things. But one of the points I was just going to say was just leading on from what you just said. The missing link at the moment is just demonstration of transmission. We've said that it's difficult to actually culture these many of these bacteria that are being collected. We need to prove that they're actually capable of causing infection. So that's one point. And then, so as I say, I was looking at cough. I'm very reluctant to say that coughing is not important for TB transmission. We published data in the International Journal of TB and Lung Disease in 2018 or 2019, linking cough frequency to positivity, IGRA positivity in household contacts in East London. But there is very little data otherwise directly linking cough frequency to TB transmission. That's one thing to say. But also regarding cough is the excellent paper by Michael Shiloh and colleagues that was published in Cell in 2020. identifying this molecule SL1 in the cell wall of M. tuberculosis. And they demonstrated very, very effectively that this molecule seems to trigger coughing in guinea pigs. And it's very difficult to explain why MTB would go to the bother of producing something that stimulates the cough reflex if it's not benefiting the organism some way. So yeah, so I mean it's probably correct that maybe in high incidence settings a lot of transmission is probably occurring without coughing, but I think in low incidence settings in particular, when you've got someone who's coughing a lot, I mean I don't think we can ignore that as a role in infectiousness.
Yeah, thanks for that. I mean, I guess I didn't mean to imply that cough is not important, because it clearly is. But you know, I've been working in TB for a long time, as I'm sure you have too, and you know, there is such an overemphasis on cough sometimes, and I think maybe that is where this might change our thinking around this. I mean, I've never thought that cough triage is a terribly good way of trying to reduce transmission in healthcare facilities, for example, and get the right people seen more quickly. But yes, it is interesting about, you know, what triggers cough. And I'm sure, and I've seen your study, yes, of course, that, you know, I mean, we know some people cough a lot more than others, and that would be interesting to sort of find out, you know, what are the factors that are associated with that, and how does that relate to transmissibility, I guess.
Katie Dale's just added that in the Victorian TB program, sputum smear positivity important. Yeah, I take a similar lesson from these studies that isn't it interesting how they're not smear positive? These bacilli found bioaerosols were not smear positive using the ZN, so the typical method. And yeah, is this method, these studies, detecting bacteria that are not adapted for transmission but may be adapted for persistence in vivo? And then, so it seems like there's something about finding ZN positive organisms in expelled air and coughing and sputum that correlates with transmissibility.
Yeah, I mean, it's possible, but it's actually not sputum that is causing transmission. I think it's important to, you know, despite the emphasis 100 years ago on people not coughing up onto the pavement, you know, it's not sputum that is being expelled into the air. We know TB is airborne. So, yeah, I think that's important to note. There is a question in the chat I see about the presence of cavities on chest x-rays. And I'm just trying to recall whether they looked at that in this paper. And I think the sample is just not large enough to have seen any difference with regard to that. I mean, importantly, a lot of there was a very high HIV positivity rate in the study one, the people who were on TB treatment or being investigated for TB, and often there are no cavities present. You know, there is TB disease present on X-ray, but there's not a lot. Cavities are not common amongst people living with HIV. And unless they're really quite severely ill when they first present for TB. So, yeah, that's also another interesting question. And the sputum smear positivity thing, you know, I mean, I think it does make sense that if you've got more TB in sputum, then you're likely to be breathing out more TB in your bioaerosol as well. So I think that's not a contradiction. Yeah.
As Sadid has said, he's asked actually, was there any demonstrable difference in the quantification of the bioaerosols between groups? So as opposed to be dichotomized, presence of bioaerosols, no bioaerosols, could they quantify the amount of TB in the bioaerosols? Helen?
No, there wasn't a significant difference. If I recall, there was a tendency for higher quantification in a subset of the group with TB, with diagnosed TB. Yeah. So they did have higher levels. But overall, not. There
was the graph that you showed where the TB category A patients had the highest number of. I think they called it bioaerosols on the y-axis compared to the other two groups, and it was the lowest group, but not by much, was Group C.
That was the percentage of the participants who had bioaerosol positive. So in terms of quantification, I didn't present those results, and they are in there somewhere. But there wasn't a significant difference, but there is potentially a difference if you had a bigger sample. I think is the conclusion of that one. Yeah.
I was sort of wondering with this discussion is whether, a lot of what we're using, whether it is the presence of a cough as a marker of transmissibility, whether it's the presence of sputum positivity, whether it's, 1 smear or 1+ versus 3+, or whether it is, in fact, cough, smear, the presence of cavities. We know all of these really are markers of burdens of disease, and just makes biological sense that if you have a higher burden of disease, of MTB that you're carrying around in your lungs, the chances are that you're going to be more infectious. You're going to transmit it more successfully to the people in your group. And I think what the work this is trying to show perhaps is whether we could get that better surrogate marker instead of looking at these surrogates. Really, can we find whether bioaerosols are the key to really determining whether there are people who are shedding sufficient transmissible bacteria in the environment? And those populations, the risk is that there are groups. Really, even in the absence of having smear-positive disease or cavities, etc., we still have a population of people that are infectious in the absence of all of this. And I think what this data is really potentially showing is that we've got an untapped group who need to have some sort of a measure, whether it's treatment or otherwise, to be able to really end TB, right? If we cannot end TB if we continue to have transmission from a group that's unknown, almost like having an animal host. Yeah.
I think it's important to note, and I'll reiterate this, that the HIV positivity in these participants is high, and in general, it means that in respiratory specimens, it's generally much more porcibacillary. So there is that. I've forgotten the second point I was going to raise now. This is a problem, Gail, when you raise too many things at once and you lose my train of thought.
Yeah,
I'm so sorry.
This whole area, it makes me think of a parallel with leprosy, though. And it's a slight counter to the point of people with more disease, more transmissible. I mean, obviously different in that TB is generally a lung infection, but there's a related organism where the theory goes that transmission occurs from asymptomatic people without clinical disease with the intermediary leprosy and some nasal infection and aerosol generation. So, and maybe there's something there, that something about fewer symptoms is accounting for a lot of.
Yeah, I mean, I think that would. Suman's put a thing in the chat about TB elimination and interventions, and to some extent, if this is true, it does explain why we have not been able to make any demonstrable dent in TB epidemics in many settings. I mean, the TB program in Cape Town particularly functions probably as well as any TB program in a high burden setting anywhere. It is relatively good. And so if there is a lot of transmission happening from people who are somewhere on this spectrum that we just are not picking up at all, then that would explain that. it does open up sort of opportunities around TB treatment for, I know there's, sort of aerosolized, TB drugs being delivered by a sort of an aerosol nebulization route. My medical terminology is not very good here, but I think you know what I mean. And I think, that is a potential research area to explore around this area. Yes, I don't like that term, super spreaders.
I know, it's horrible. I'm sorry.
It's not a very nice label to put on people.
No one wants to be the super spreader. Richard, go ahead.
So I was just going to go back to your point, Gail, about how we detect who's actually spreading this TB around. I don't know, so I missed the first part of the talk where you mentioned on the Catherine Williams and Mike Bearer's papers about the mask sampling studies, where there's a sort of adapted surgical face mask that's got a gel that captures MTB, and then that can be checked for PCR. I think that the most recent studies from that group, I think, have been able to measure, to quantify TB DNA in these face mask samples, and then link that with infectiousness. So that seems a sort of a simple solution.
I mean, that would be an ideal way of measuring this, a much easier way of measuring this. My understanding is that some of those studies have not been easily reproduced because, so it's the face mask technology at the moment is not really sensitive enough to really pick up, enough bacteria that we would be able to measure this properly. So certainly as a diagnostic, I don't think it's quite there yet. And probably for this method, for research methods around these questions, it's not there yet. But that would be great, much more simple way to do it. I see this very long message.
Katie, I think, Katie, you're welcome to turn your mic on. And,
yes, please.
But if she can't, I think what she's trying to say is that, in the resource setting that we have, we need to prioritize those that we know for sure have transmissible infection, which I think is a good point. We know what we know, and even if some people may not be transmissible today, it's also important to target them, because obviously that disease could progress. I just think we really don't know as much as we do about TB as we should. But on that note, I think we should wrap up because we're at 2:00. But I would like Helen to tell us a little bit about the study she has planned. Helen, you mentioned it right at the beginning, I think, in terms of.
Oh, well, it's not just me. I'm one of many investigators, and Suman, who is on this call as well, as one of them. So we have a little grant to set up some laboratory-based studies looking at the impact of UV irradiation on nebulized TB inside a chamber. Basically, we want to be able to see how much you can kill TB with UV irradiation, which hopefully is the start of a whole series of potential research work that we could do around infection control for TB, which would be really important moving forward. Very briefly.
And thank you so much to Helen. I think you've stimulated a lot of thought, and slight concern that we may not know much about how TB is transmitted as much as we thought we knew. We knew even less. That's wonderful. But thank you so much for the wonderful summation of those two papers. I really appreciate it.